Assalamu alaikum. My name is Khurram Bashir, and my expertise are in plant biotechnology. And I have recently joined LUMS after working around 18 years in Japan. And I am introducing my project related to drought and stress-resistant crops. And our aim is to work on cotton and the traits we are looking for the drought and heat through automated phenotyping screening through automated phenotyping system. We all know that the plants are grown in the field and where they face several challenges, for example, high or low temperature and the drought, I mean, water shortage, that's very important. And we know that due to global warming, in future, heat spells and drought stresses would be more recurrent and more frequent. And there are many other abiotic stresses that also affect crop production. So I'm not going into those stresses, but that are also important, and we might look also for those things. And that this is directly related to sustainable development goals, that is zero hunger. And through agriculture development, we can eradicate hunger, and we can also contribute through such to poverty reduction. Okay, and we would be looking screening crop plants through based on the leaf temperature for drought and heat. And the plants may look hot, and they may appear cool, and that's very interesting. And I would simply explain that the plants, when they appear hot, they are most probably they are would be would be drought tolerant, and when they look cool, they would be heat tolerant. That is a very simple phenomena. Plant transpire water as a process of during their development, and by regulating this transpiration, if they reduce the transpiration, the leaf temperature increases, and if they increase the transpiration, the leaf temperature drops, and that gives them the ability to withstand different kind of stresses. For example, this is my personal data. I have been working in this field since long. So when we compare, for example, A and B, for example, here. So in A and B, when you compare, so you can see that in B, the leaf temperature is higher compared to the A treatment. And these plants are performing much better when they are subjected to the limited water supply. So this criteria can be successfully used to screen a large pool of crop plants. And our objective are very simple. We want to establish an automated plant phenotyping system or screening facility, and then develop a mutant library of cotton seedlings, and then screen these mutant based on leaf temperature as shown in the previous slide. And when then characterize the molecular mechanism, we will see how uh, they are regulating the transpiration or leaf temperature, and this would enable us to develop our identification of plants that are performing much better during heat and drought tolerant stress. And we are looking for to establish an automated phenotyping facility to screen a very large pool of crop plants. And this would be the first of its kind in Pakistan. And this would enable us to maintain a very homogeneous environmental conditions. And when we grow plants in fields, there are macro and micro pockets which uh, with varying temperature and humidity, which might affect our screening process. So we would be establish a very robust and automated system to screen plants, which would be recording data every 3 or 6 hours for various characteristics. And although our application for a GCF grant was not successful, but we have not given up. We are actively looking for funding, exploring different uh, opportunities such as JICA through ODA. But what I want to emphasize that our idea is very simple, and we are looking for the funding.